Good afternoon, everyone. Welcome to our Next Gen Omics series webinar. My name is Dan, and on behalf of Oxford Global, I would like to thank you all for joining us today. Today, we will have Paul Monnier and Maria Miller from Cytosurge, who will be presenting on FluidFM, a new approach to CRISPR gene editing. After the presentation and demonstration, we will have time for a short Q&A session. So please submit your questions through the chat option on the bottom of your menu display. The full recording of the webinar will be made to download on our secured website, and the details will be sent to you after the webinar. So without any more delays, I would like to get started with the presentation. So firstly, Paul, over to you.
Hi, hello, good morning or good afternoon, everyone, depending where you are in in the world. This new FluidFM webinar. Today we will talk about a new approach that we propose to CRISPR gene editing with FluidFM. So first, let me present you a bit how we will organize this webinar. I will present quickly the technology. As I noticed that we have a lot of new people that registered. We registered and we never heard about FluidFM. Then I will talk a bit more about injection and cell isolation with FluidFM, and later how we can apply that to the CRISPR field and how we could improve, how we believe we could improve the CRISPR field. Before that, Maria, who is in the lab currently, show us a live demonstration of the system injecting CRISPR complexes into CHO cells. So what is FluidFM? FluidFM stands for fluidic force microscopy. It's a technology which is derived from atomic force microscopy. The innovation that we have made at Cytosurge is that we have a channeled probe, the hollow probe, connected to a pressure controller, so we can pull liquid through the probe. A really important part of the technology is the probe, and we have different tips at this probe, allowing different application. With a flat probe, for example, we can do cell isolation, measurement of adhesion forces, or if we use a nano syringe with a pyramid and a sharp apex at the end to be able to penetrate the cell, we can perform injection into cells or even extraction from single cells. We also have what we call the FluidFM nanopipette, that we can use for spotting or nanoprinting in, for example, micropatterning applications. So today, I would like to quickly present you a single cell nano-injection. We have previously already made some webinar about it, and I suggest you to go on our website if you want to know more about it. But I wanted to explain it again a little bit, as it is a core FluidFM method regarding the CRISPR application. So for single cell nano injection, we use this FluidFM probe, the nano syringe, which is really sharp. We notice that it's really important to have a very sharp apex here to be able to penetrate the cell really gently, and not damage it, so the cell can survive after the injection process. And the aperture here is around 600 nanometer. In size, it's vary a little bit from batch to batch, but it's always around 600 nanometer, which allows to pull, to inject liquid and complexes, protein from any kind, that any soluble compound that will be smaller than this aperture. Here we show it was done by Oram Guillaume-Gentil in 2009, who developed the technology to perform injection. And she was able to show with the laser feedback of the system that you can measure the perforation of cytoplasmic membrane as well as the nuclear envelope. And like that, she was able to show that you can use FluidFM technology to penetrate cells and inject here, for example, a red fluorophore, it was rhodamine, together with a plasmid, a GFP plasmid. And after 2 days, the cell was still viable and expressing the plasmid. It was the first experiment, the first proof of concept that FluidFM could be used to inject adherent cells. Here is a little video to show you how the system works. So this is our setup. This is our FluidFM BOT system mounted on an IX83 microscope. And I wanted to show you this animated video to visualize a bit more how it's performed and how it works. So first, thanks to our really nice software, you can just point and click the cell you want to inject. And by giving a few parameters, you will see later in the live demo that Maria will conduct, then the system performs the injection autonomously, thanks to this laser force feedback, which is let's say at the core of the atomic force microscopy technology. Here, it allows the system to know when it penetrates a cell, the probe will slightly bend, which will deflect the laser, and therefore tell to the system that the probe has penetrated the cell. This is what allows automation of the injection process. After the injection, you will see during the live demo, the system records the position of the cells that have been injected. This is really useful if you want to come back to this cell later on for re-injecting, for example, or isolating, or if you also just want to do long-term observation of the cell. So often people ask me in what cell type can you inject? Have you tried with my cell line and etc.? So I cannot say that we can inject in all cell lines, but so far in every adherent cell line that we have tried, we could figure out the right parameter to inject properly. Here is a few examples with C2C12, there are myoblast cells, or for example, human embryonic stem cells, or mouse embryonic stem cells, pig neurons, or other cell line. So far, every adherent cell lines we have tried, we could figure out some parameter to inject. You can play with the system. You will see later on with Maria's demo. You can play with the pressure, with the approach speed, with a few parameters that are really easy to handle to be able to successfully inject into your cell. Usually, we reach quite efficient. We have a quite efficient success rate of injection, above 90%. It obviously depends on the cell types. And what I mean success rate, it means also that the cells are viable after the injection. Not only that we have injected them, but that they survive after the injection. Another question is what kind of compound can we inject into these cells? In theory, any soluble compound can be injected. Depending on the viscosity, of course, it will be more or less difficult to inject it into a cell and keep it viable. But so far, everything we have tried, from fluorophore here, like lucifer yellow, or proteins like UHRF1, which is a nuclear protein, small RNAs, plasmids, CRISPR-Cas complexes, RNP complexes, or even big protein like antibodies, we could inject it into cells. Here it's interesting to know on the two upper left pictures that we can also distinguish if we inject into the nucleus, like here the UHRF1 protein, or into the cytoplasm. You can really select where you want to bring your compounds, which is quite relevant depending on your application. Sometimes you want your compound only in the nucleus or only in the cytoplasm, depending on what you are studying or what you are doing during your experiment. In terms of viability, we have a quite high rate of viable cells after injection. Here is an example of CHO cells that have been injected with lucifer yellow. After injection, after 5 minutes after injection, I stain them with propylium iodide. The specificity of propylium iodide is that it will penetrate the cells only if the cell is dying, because when a cell is dying, the membrane becomes porous to the cells. And we could notice that after injection, we have like that up to 95% of viability, which is really good. It obviously depends on the parameters that you use during the injection, and also, of course, the cell type. You have to play a bit with these parameters, but it's quite high rate of viability. Another advantage of using propylium iodide to stain to check viability is that I can also check that the membrane is resealed after the injection. Indeed, when we penetrate a cell physically, it creates a hole into the cells. And if if I, for example, use a propylium iodide during the injection process, all the cells will be stained in red because there is this hole in the cell membrane. And like that, I could notice that after 2-3 minutes of the cells, after the injection, the propylium iodide does not go inside anymore, meaning that the membrane is really resealed after the injection process, confirming that it's a really soft process. Why people use nano-injection? It's really relevant when you have rare or precious cells. You cannot afford to use transfection method that will kill millions, hundreds of thousands of them. That's why some of our customers use FluidFM. For example, here, injection into cardiomyocytes that was performed in PKU, in Peking University in China by one of our FluidFM users. Interestingly, he could observe that using our probe to inject, thanks to the laser thought feedback, he could also measure during the injection the beating of these cardiomyocytes, which could be also a quite interesting application using FluidFM. A really cool feature of FluidFM is that we can also measure the volumes that we inject into each cells. How do we do that? We use a really high precision in the microfabrication of our probes to use the probe as a reference for a given volume. I mean that when we inject a fluorescent compound, we know that the probe has a specific dimension and we use it as a reference for fluorescence intensity, and then we can compare it to the cell intensity after injection and calculate like that what volume we have injected. We will see later on during Maya demo how we do that quickly. We also made in the past a webinar dedicated to volume measurement with FluidFM. I encourage you to visit once again our website if you want to watch this webinar. To explain you quite quickly, but Maya will also show you that during the demonstration. We have implemented into our software this volume calculation tool where we use the probe here as a reference. We take into consideration the background, of course, to deduct that from the intensity of the probe, and then select each cell that we have injected, and like that also deducts from that also deduct the background, and the system then calculate what volume has been injected in each cell. Here, for example, a few femtoliter, around 100 femtoliters in most of the cells that have been injected. The second core method I wanted to mention regarding FluidFM applied to CRISPR technology is single cell isolation. Here is the principle of FluidFM for single cell isolation. We use a flat probe that we can then approach the cells, apply a negative pressure to pick up the cells, and then move it to another well or another substrate to deposit with a gentle positive pressure. The really interesting thing with FluidFM is that we can also isolate adherent cells. If we fill the probe with trypsin, we can target the cells of interest, gently deliver trypsin with a really little positive pressure that will detach the cell of interest. Then with the same probe, approach this cell, apply negative pressure to pick it up, and go to another well to release these isolated cells. Here we have a little animated GIF that we show we can, with the software, target the cells and the probe approach it, what we call our FluidFM micropipette, and then pick the cells by applying negative pressure. Then we can select in our software in which well plate we want to isolate the cell, and the system will go there automatically to the new well, and then approach the surface, the well's bottom, to gently release the cell. The probe will move on the right here, and you see that the cell has been released. We also reach quite high viability with single cell isolation, around 80 to 90% of cells that survive after this process. And now I would like to talk with you a bit more about how we think this core methods, single cell injection and with volume measurement and single cell isolation, could to our sense, improve the CRISPR field and single cell gene engineering. So one of the main issues in the CRISPR field is delivery of CRISPR complexes, and especially delivery to the nucleus. With FluidFM injection, we can really target the nucleus on any adherent cell and bring the complex definitely where they are needed, independent of the cargo that we use. So we can inject the CRISPR-Cas RNP, ribonucleoprotein complexes, the plasmids, or even the mRNA version of the CRISPR-Cas components that you need, together also with the HDR repair templates. Our process is also highly automated, so you can, like that, inject hundreds of cells per hour and quite easy to learn. You will see when Maya makes the demo, it's quite easy, quite straightforward software that we have developed with straightforward workflow to inject into cells. So regarding intranuclear injection, so as I mentioned, we deliver directly into the nucleus. Here is an example of a picture of an experiment I did with mouse primary hepatocytes, where I injected the two nuclei of this. These cells have the particularity to have two nuclei. I injected both nuclei with CRISPR-Cas RNP complex. We really believe that by bringing all the complexes together at one in the nucleus, we can improve efficiency of CRISPR editing, especially when we want to do precise editing and you need a repair template. Quite often, this repair template risks to be degraded in the cytoplasm. If you use normal transfection method, you don't really control what amount of these molecules will penetrate the cell. So together with the control of the amounts that we deliver, because we can calculate the volume of what we inject, we really believe that we can increase CRISPR efficiency. And we are working together with academic lab right now to prove this message. Let's say the preliminary results are very encouraging towards that point, and I hope we will soon be able to communicate them. It's particularly relevant to use CRISPR to inject when you work with difficult to transfect cells, like embryonic stem cells, but also neurons. For example, here, an experiment that we have done in Jinan University. Last time I visited China, I could perform plasmid injection into neurons. It's really well known that as these cells do not divide, bringing plasmids inside nucleus of these cells is really, really difficult. But with FluidFM, we could reach quite high rate of transfection efficiency. Another advantage of FluidFM is what we call multiplexing. So when you inject a CRISPR-Cas complex into a cell, it's to target a specific gene to induce a mutation. What people more and more try to do is to multiplex this editing, meaning target several genes in the same cells. Currently, it's very limited by the delivery of the complexes. It's really difficult to transfect with a traditional transfection method to transfect a lot of guide RNA that will target many genes. And also for other reasons like cytotoxicity, it can become really, really tricky. It is more and more, though it is more and more interesting for people working especially in multigenic factor disease, in de-extinction projects or in genome writing. There really needs to induce hundreds, if not thousands of mutations at once in the cell. Here, FluidFM has a really advantage, as we can load, you will see later during the demo, on nano syringe, we can load it with 1 microliter of any compound. And this microliter, you can bring as many guide RNA as you want at once, therefore really improving the multiplex delivery of CRISPR-Cas complexes, whether it is the mRNA version or RNP ribonucleoprotein complexes version. Sometimes it's not possible to modify hundreds of genes at once because it will induce DNA damage response into the cells, and people would favorize what we call stack editing. Stack editing means that we repeatedly modify the genome of the same cell at different times. And with FluidFM, as you will see later on during the demo, we can record the position, the coordinates of each cell that we are injecting. Therefore, it's really easy to come back to the same cells a few minutes or a few hours later once the cell has recovered to perform this stack editing and therefore perform serial injection into the same cells with different guide RNAs. Another interesting aspect is to use FluidFM to optimize efficiency, the ratio between efficiency and off-target via our precise dosage that we can perform via our volume calculator tool. What is off-target in CRISPR is when you target a specific gene with your CRISPR-Cas complex. Sometimes the Cas9 will also cut at a different location and induce unwanted mutation. It is well known that it is a stochastic process that depends on the exposition time and the Cas9 availability, though there is currently no other method that allows to calculate precisely what amount of CRISPR-Cas material you bring into the nucleus. So we really believe, like here, for example, on this picture, you can see a full colony of egg cells that I have injected with CRISPR-Cas complexes, and I could calculate the volumes that I've injected in each of these single cell, and therefore I can relate that to the concentration of Cas9 that is in each of these single cells. Like that, if I combine with cell isolation and then perform further analysis on these cells, I can really link it to the off-target effect, that I will observe to the concentration that was induced. Another advantage is as we isolate the cell, we can isolate the cell right after the injection. There is no division of these cells. It means that you don't need to wait the cells to divide to have enough material to do your analysis. So why is it a problem? Because when you have division in a cells, you have random mutations that appears. And then when you sequence your genome, you don't know if these mutations that you see is due to the off-target of the CRISPR or to a random mutation due to cell division. So for all this reason, we believe that we can use FluidFM to really optimize the ratio between efficiency of CRISPR and off-target effects. Finally, before we move to the demo, I wanted to talk to you about a really nice application, about nice results that we got recently into the lab that illustrate a bit what I talked a bit earlier, especially regarding multiplexing and efficiency of CRISPR. Here, I try to perform a triple knockout in cells, in CHO cells, to create some cell lines, monoclonal cell lines using FluidFM. So what I did is that on the first day of my experiment, I injected 3 different guide RNA together with Cas9, together with GSP mRNA and Lucifer yellow in CHO cells. The Lucifer yellow, I used it to monitor the efficiency of my injection, making sure that I really inject into the cells. Then on the next day, I could observe the expression of the GSP mRNA that allowed me to go with a micropipette, with a FluidFM micropipette to isolate these cells that I knew I really injected the day before. And then meaning that I knew there was CRISPR that came inside and that they were still viable. Then a few days later, all my clones could grow and start to create colony. An important fact here is that we are sure that we have a monoclonal cell lines, because when we perform FluidFM cell isolation, we visually observe that we take only one cell. So it really shortens the time generation of monoclonal cell lines compared to other traditional techniques. And then I let them grow a little bit more. After 10 days, I could start in my analysis and realize that 50% of my clone had the 3 mutations, which is a really, really good ratio in terms of multiplexing CRISPR editing. And then after 14 days, I had enough material to really say that I have my monoclonal cell line and I can use them for downstream application. So before we switch to Maria for a demo, I just would like to remind you or inform you that we are hosting a FluidFM user conference. If the COVID-19 situation does not get worse, but it should be in the end of June, so it should be fun, and we will open registration soon, if not done today actually already. So you can visit our website on this URL register. This is a really nice conference where we invite our user to present their data, their results, and we also invite our curious people who like to know what to do with FluidFM, what works, and we will like that to really strengthen our community of FluidFM user people. And last year was the first time we made this conference. It was really high level of scientific communication, and we were really happy about that. And we are doing that again this year, then in June, and we really encourage you to join us in Zurich this end of June, beginning of summer. So now I finish with my presentation. I will hand over to Maria for a live demo from our lab in Zurich, and see you later for the questions.
Hello everyone. I don't know if you can see me. I hope yes. So my name is Maria, and I'm here at the lab that we have at Cytosurge in Zurich. And well, I'm gonna try to show you how does this system works in life, the system that Paul introduced you now. And for that, we're gonna do a small injection experiment. We're gonna try to inject CRISPR-Cas9 tRNA into cells together with a fluorescent lucifer yellow. So then you'll be able to see all what Paul was saying. Before starting, so then just you can make an idea of the things that Paul mentioned. So we're working with nano syringes that Paul showed in the presentations. These nano syringes are glued in this small consumable. It's a bit difficult for you to see it, but on the very end, like here, yeah. So you see like a small black dot. This is the cantilever that we're going to use for our experiment. So this consumable, we are like selling them in this kind of package, which contains a QR code here in this side and some information regarding to the probe that you're having. And with this QR code and the reference number that you have for each probe, you can know some specific properties of each syringe that you're using. So that's what I did already before the experiments. I just put a plate of CHO cells inside the device. And then also what I did is just prepare, I mounted this consumable, this plastic consumable that I just showed you, I mounted it already inside the bot. So now without no more delays, I'm gonna start showing you how this works. So I'm gonna switch and show you the screen. I hope that everyone can see my screen now. If not, please let me know. So this is the software that we're using. It's called ARIA, and it allows us to control the system in multiple ways. So from one side, we have this navigation tool that allows us to navigate inside the wells of the plate or the Petri dish that we're using. We can also take pictures or videos. We can also control the pressure that we are applying to our probe, like to this microfluidic system. I'm gonna just change the pressure now. We can also measure like the movements of this cantilever, which is the pressure, which is the mechanical pressure that's like doing this probe on the system. We can also control the movement of the probe if we're like touching the surface, if we want to retract it, or if we want to approach just softly. We have also washing tools and also visualization presence tool. So now what I'm gonna do is I'm just gonna go to the well where we have the cells and turn on the bright field so you can see how is this going. And so I will remove some of these tools which we don't need. So here on the screen, we have, this is our cantilever. That's the image, actually that's the proof, the images that, that Paul showed you before. In this case, it's a nano syringe, and you can see like the sides of the pyramid that's on the very top of the cantilever. On the bottom, we have the cells. I just see them in a very low confluence, so we could see better probably the injection. And then actually that would be good in case you would like to do some analysis on the growth when you're injecting a specific compound or something like that. Then you let the cells grow without problem. So what I'm going to do now is I'm just going to easily select the injection workflow here. And I'm going to tell the system what I want the injection to be done. For example, let's select this cell here, this cell here, and this cell here. You can see that you can really easily move this crosshair around, select the position, the exact position that you want your cantilever to go, so the action to be performed. So it's really, really easy to use. Once we have the list of points that we want, we just click on OK. And now we can select the different injection parameters. For example, we can change the set point, which means that this is the like the force that the system will do once it will touch the surface. We set up like a 250 millivolt, which is not a high, like a high force. We also can play with the approach and the retraction speeds. So how fast or how slow the probe goes to the cell and releases like the cell as well. We can play with the pressure, which is really important because some compounds that we try to inject might need higher pressures. For our experiment today, I think that 70 millibar should be more than enough. You can also play with different injection times. 3 seconds must be okay as well. You can even go up to 10 seconds and, for example, inject slowly. And finally, you can also play with the retraction distance or the idle pressure. But for this case, we're just going to modify the idle pressure, but it's the pressure that the system will keep once you have injected. So in between the injections, let's say. So 5 millibars should be enough. So now we're just going to click on OK. And the system should go where I pointed. If not, we're going to see. You can see maybe the pressure is too high. Also, during the process, you can always know how long this will take, more or less. There's a timer right next here on the right side where I'm pointing. So you can have an estimation of the time that the full workflow will take. For example, if you plan to inject 200 cells, of course, you won't be all the time in front of the computer. So then you can have an estimation of the time of this process. So as Paul mentioned, once the injection is done, the system keeps the position of all the cells that have been injected. You can name this. For example, we're going to call this 2020-03-19 injection 01, like as a group. You could even put something like, for example, like your compound concentration, or even the type of cells that you're working with. So we keep it safe. And what I'm going to do now is try to check which cells have been properly injected with the fluorescent compound. For that, I'm just going to remove this cantilever because the plastic of the consumable can give some background. Like this. And I'm just going to turn at the same time the filters to visualize them. And here they are. So if you want to see exactly which were the injected ones, here you have them. Maybe we could try, now that we said just 4 out of these, just 2 of these out of these 4 cells were perfectly injected. What we can do is just try to repeat it again by modifying the the parameters that we have applied. For example, I'm just gonna move again back here. Sorry, here. Move to bright field again. And for example, move here that there are not that many cells and restart again. Just going to modify the pressure that we applied and the injection time. And the system, as you can see, will go automatically to the position that we set. And inject the compound. I don't know if you could hear it, but there was a sound just right after the injection. This sound corresponds to the release of the pressure from the pressure control system. It's totally normal. So actually it means that the system is working fine. And yes, and now just keeping injecting. We can also follow the process with fluorescence. Now. Maybe too much for this one. So you can see, especially with fluorescence, you can see how the cantilever approaches and then it retracts as it loses the focus, like now. So now we have again the 6 positions that we checked. I'm going to retract again the cantilever so you'll be able to see it better. And I'm going to give again another name to this group of points. And what I would like to show you now is, like with this group of points, how can we measure how much have we injected? So for that, what we have to do is we can take a picture of, we have to take a picture of each point by using the observation workflow. In this list of applications that we have on the right, as is observed, we're just going to click it and select these points that are this group of points. That's the one that we've just done now. Just going to click on OK. And what we're going to do is just, we're going to record an image. The cantilever must be retracted during this time. We don't want the cantilever to be closed because otherwise we can have some background interferences. Then what we're going to apply during this time is like these current settings. But as you can see, you could just create your settings and then just select the ones that you want. Another thing that you can do is like in case you have, like, for example, multiple observations, imagine a picture every hour during 16 hours. So you could select also what's happening to the microscope during this time. So what we're going to do is just put idle, which means that there's no illumination. But imagine that you would like to have your cells with a bright field. You could also select it here. And in this case, since we're just going to take one caption of each cell, I'm just going to put like 1 repetition, but you could repeat it, as I said before, like for example, 16 times, and then you could take a picture every hour. Since there's going to be no repetition, I'm not going to change also this minute time here. So I'm just going to click on OK. And now the system will center, as you can see, each injected point in the middle of the screen and took a picture of each one. You saw it's quite fast, but like this, we make sure that we have all the points like in the center of the image and like with the configuration settings that we gave. And now we have these pictures. What we can do is open the volume calculator tool that's here on the left menu. And first, what we need to do is to give the reference. So actually, Paul mentioned it before. We need to give like a fluorescent reference, so the system knows more or less like the system can relate the fluorescence interference with the volume. For that, we use the top part of the cantilever. So I took a picture shortly before the demo of this cantilever. It's in the same, actually, this one is exactly in the same preset, visualization presets that the pictures that we took. And we have to tell the system exactly which is the part, which part of this cantilever we'll use as a reference. For that, I'm just going to first select the background, as is it said here, first background, for example, this. And now we're going to just choose this upper part of the cantilever. So our cantilevers, these probes, are coated with a laser deflective gold layer, except this part on the top. So that's the part that's interesting for us for the measurement of the volume. So we're going to apply the reference. And once we have selected these two compounds, what we're going to do is just to go to the cell image selector. Here there's going to be like all the group points that you have in your system, and the ones that you have pictures of. That's why we took the pictures before with the observation workflow. So I'm going to click in this one, and I'm just going to select all the pictures, and we're going to click on apply selection. And now we're going to start with the image processing. So as we did before with the reference, we also have to select the background. Here we have. And now we're going to select the area, more or less, of the cell that we want to measure the volume of. For example, this. We just click on apply. And now here in properties, there's going to be like all these parameters. So basically what it's telling us is like which area have you selected as a background, which intensity had this area, and also which is the area that you selected as a cell, as a sample, and also which intensity does it have. Then it will compare this with the reference that we have, and it's going to give like a measure. For example, in this case, it says that it has this volume. We can go to the next one. I'm going to do the same quite quickly if I manage. Yes. No. I'm not sure that's the center one, but this is one. Okay. And for each one, as you could see, we could get the volume. I'm just going to stop here. If not, it will take quite a long time. And once we have done this with all the cells of interest, we can click on report, and we're going to have the information of each point, also in which group they were, like these cells. So you can know also which conditions you had there directly by checking this table. And then you can know which is the volume that you have injected in these individual cells. You can, of course, save it. It's always telling you in case you haven't done all the images that there are some missing. We're going to save it anyway. And then here in the results history, it will appear here on top. So as you can see, then you can export it and do with it whatever you want. You can export it actually to process the data on Excel, for example. Then with every picture, you can also like work with it. You can export it as well. You can also export the metadata of each picture, and you can also save it wherever you want. And yeah, that's basically how our system works. What I'm going to do now is I'm just going to approach again to the sample, and I'm just going to leave the system running. So then just feel free to quickly ask for any question that you can have. And either Paul or me, we're going to answer any question that you can have. So that's it.
Thanks to both Paul and Maria for that presentation and demonstration. That was really interesting. And so now I'd like to open the floor for discussion. So remember to send us any questions you have using the chat feature of the platform. and I will read them for Paul and Maria to answer. So we have a first question, and that is, how do you make sure that the compounds are delivered into the nucleus?
So maybe I can answer to that question. It's thanks to the force feedback system. So the probe can really know, the system can really know when you have perforated the cytoplasmic membrane and the nuclear envelope. This is the theory. In practice, we go to be really fast and have a higher throughput. We go really fast in each cell with a high set point, high force feedback. So we are sure we really penetrate every cell, and we feel when the probe touches the bottom of the well below the surface, and as the opening of our probe is on the side, we are sure that every time we touch the bottom of the well plate below the cell, that the opening is inside the nucleus. And we select really XY, as Maya showed you, to be on top of the nucleus, and then it approaches vertically to goes down, and like that you can make sure that you really deliver your compound into the nucleus if you want to inject into the nucleus.
Okay, thanks, Paul. And the next question is, what cutting efficiency do you have with FluidFM when injecting CRISPR?
What cutting efficiency? I mean, we didn't precise that we do a specific anti-fouling coating on these probes before we perform injection. So we are sure when we penetrate the cells, there is no debris from the membrane or from inside of the cells that will stick to the probe and that will clog after a while the aperture. It's really, I mean, it's really obvious when we don't perform the coating after 20, 30 injection, the probes start to get clogged. But with our specific anti-fouling coating, we can really inject thousands of cells without issues on clogging the probe.
Okay, and can you perform CRISPR injection into plant cells?
That's a good question. We never tried so far into plant cells. There is some. It's a question a lot of people are asking because it's really well known that plant cells have some of the species are really difficult to deliver with CRISPR complexes. We have the. I mean, we believe it should be possible, though we don't know yet how exactly in the procedure. We believe if we could approach with a probe with a specific cocktail of enzyme to, for example, digest the cell wall, because this is the main difference between plant and mammalian cells. They have this really thick cell wall that you need to get read through, so our nanosyringe can go through. We believe that it will be possible, and some of our users have asked us the question already, and it's not impossible that some try some things about that very soon.
Okay, fantastic. And so next question, is there a way to inject all of the cells of the well without individually clicking on each one?
So, well, at the moment, we don't have that option. So you have to really select the cells that you have, but we have in mind to optimize the system with artificial intelligence. So you have to tell the system with how the cells look like, which is the center of the cell, which is the point you want to inject into your cell. And once you have done like all this training to your system, let's say steps, then you could probably inject all the cells. At the moment, it's not possible, but I do agree that's an interesting option because then it even saves you more time.
Okay, and this next question, I believe, was addressed to part of your presentation, Paul. And it's, have people tried to do mouse zygote injection with this approach?
Excuse me, can you repeat the question, please?
Yeah, sure. Have people tried to do mouse zygote injection with this approach?
Mouse zygote injection. So not yet. It's a bit like plant cells have a really thick protective layer around it. I would like also to try. For now, it's not yet. I cannot say it's possible because we never tried. No one ever tried. But some group who have our system already, I know are working. We're working already before on zygote injection, so I really hope that they will try and we could get some nice information about that soon. But But so far, there is still this question mark about the zona pellucida around the zygote. That could be an issue to be able to really penetrate through the membrane. But that's a really interesting application that we would like to see in the future.
Okay, fantastic. And so for the next question, I assume you need some special treatment of the probe before filling it with DNA and proteins, so the nano outlet does not get clogged. So what exact treatment do you use? Yeah, we'll stick with that first part of the question.
Okay, so then I will go for this part then. So basically, as Paul said before in the previous question that we had, normally what we do is we coat the probes to not have issues with the cell debris when we're injecting into different cells. Of course, there are things that we cannot overcome, like in terms of clogging. It means that if you're working with a really highly concentrated solution, the probe will clog anyway. It doesn't matter what coating you're doing, which treatment, and so. But once you have optimized this concentration of compound, you can perform, I don't know, a coating like, for example, with Sigma coat, or also with derivatives of the PLL like Pomoxa. And with this, what we have seen is that we don't have issues with the cell debris that put attached onto the cantilever, and therefore we can do multiple, like hundreds of injections with the same probe. I hope that answers your question.
If I may add also a little detail, but when we prepare an experiment, we also filter usually the compound through 0.2 micron pores. This is not possible, of course, for every compound. It depends on the quantity of compounds that you have, but also for proteins, for example, that tends to aggregate. We have developed some protocol. We really are careful. We centrifuge the sample in case of aggregate to really take only the supernatant to make sure that we don't take aggregates that will clog the probe. But we are really developing protocols for every kind of application. We have some already available on our user center on our website, and of course, yeah, it depends for every new compound might require a little bit of adaptation of the protocol.
Okay, and did you try with suspension cells? And if not, do you think it would be feasible?
So we have tried, not exactly suspended, but we have tried to isolate easily, like suspended cells. Like you have your cells, you have trypsinized them, and you have like all the cells suspended in a solution. We could do that without problems. We fear that with the injection, for example, the cells will just move as soon as you approach them, and therefore we are thinking to find some ways to attach the cells on the surface, at least temporary, so we can perform this injection as well with these cells. We think it's possible. I mean, it shouldn't be a problem. There are plenty of methods where you can temporarily attach your cells on the surface. So I think that it would be easy to develop.
Okay, fantastic. Also, regarding injecting HDR donor template, which maximum size did you try? Do you think it would be feasible with large template, 2 to 3 kb?
2 to 3 kb, I guess it's definitely possible. As I didn't try with the repair template, but I tried with plasmids that were much bigger than that. TNA tend to stretch, even if it's too big for the opening, like big plasmid, they usually tend to stretch when you apply pressure. So in theory, it's possible. Like so far, I tried only with a really short donor template, but I don't see any reason why a 2 to 3 kb templates will not work. It might be a bit more viscous, the solution, if then you have a higher concentration of DNA, but not such viscous that you could not inject. So I don't see any technical reason why it should not work.
Okay. Wouldn't it be better to inject while in fluorescence to check if the cells are being injected or not before the end of the process? And secondly, how can you know without fluorescence if the injected volume is getting inside the cells or not?
So without fluorescence, we cannot know. That's sorry, maybe I was not clear when I presented that, but we always use fluorescent compound if we want to measure the volume. Mm-hmm. Then, of course, sometimes for some application, it might be not possible because you might not want to have fluorophores together with your compounds. And for that, what we advise is to first inject both your compounds with fluorescence, and then you will have an average of what you inject. But then you lose a really single cell calculation, but you still have an average of what you inject into the cells.
Okay, fantastic. And are the injections parameters different depending on the cell line?
Yes, indeed. I mean, even it's not just on each cell line, but also sometimes even in each cell. So you can really see, I mean, for some cells, you have to really be careful because they can detach even when you're like retracting your cantilever, because the way they are attached on the surface is not that strong as others. There are other cells that you have to inject them when they are like more dense. For example, today it was quite low density, but we saw differences also on the injection success, depending on if the cells are really confluent or if it's just like in this case, like a 40% confluent or something like that. So therefore, what we always suggest is like to make a first trial with your cell line, like in the same conditions that you would like to run your experiment. So then you can have more or less like the parameters that you will use for the experiment. We always advise this.
Okay, fantastic. Thank you very much, Paul and Maria. I'm afraid that's all the time we have for questions at the moment, so I apologize if we ran out of time for me to put your questions to the speakers today, but I'm sure that if you get in touch with Cytosurge and Paul and Maria could potentially answer those questions you might have, or if you have any further questions, then please feel free to direct them through us through the email on the screen at the moment. So thank you very much, Paul and Maria, for the excellent presentation. I'm sure everyone enjoyed that, and on behalf of Oxford Global, I would like to thank everybody for joining us, and thank you to Paul and Maria for the time and effort you put into the presentation, making it as interesting and valuable as it was. So yeah, as I said, if anyone has any further questions, please do email them across to my colleague Bradley using the details on the screen. And all that remains to be said is have a great day, and we look forward to welcoming you to one of our future events. Thank you very much.